How can we get journals on board with allowing replicated studies to increase overall numbers? And that was a question brought up during the why standardize discussion.
Could you repeat that one more time?
So the question was, how can we get journals on board with allowing replicated studies to increase overall numbers? And I'll let anyone who wants to get started with that one to kick off.
I guess no one wants. I think it's a very difficult question. And for example, I myself had an experience for a manuscript that was published last year that we wanted to report a new parameter, and we compared to other parameters, for example, also the proliferation markers. And one comment from a reviewer actually was, that this is common knowledge, and we basically just added this as supplemental material just to see, look, our data is representative, those parameters work, and the reviewer was like, this is common knowledge, and we don't need this information. And I don't think that's true. I mean, like all the studies that we have include small case numbers, only one pathologist, with all that variability that comes into place. We have different outcome assessment, all that. So we need studies that confirm that, and especially I think we should encourage studies that wants to, for example, evaluate a new parameter to also show that the other parameters are working in your quote-unquote working in your study population, to first of all show that your study population is representative, and also to show reproducibility of that. So I think there needs to be changes not only in journals but also from reviewers, which is kind of arbitrary because who's doing the reviews, but that should be encouraged, especially if only been reported in supplemental material. But this is important information still for meta-analysis and systematic reviews if you have this information reported as supplemental material, for example.
Is there anybody here from a journal who's an editor or a subsection editor or anything like that from JVDI or VetPath or VCO or Compath or anybody?
Okay, well, I'm a new associate editor of the Image Challenge, but so that doesn't really count. So the question is in terms of studies that validate other results. Was that the question? So
the question was about how do we get journals on board with this, and what is that, what is that process like?
Yeah, so I know that Don wants to write letters to journals. And I know that he also wants to create a summary of some of these guidelines that's, captured in a small, in a page, so that, journals can have just a quick reference to look at in terms of some of these standardization parameters. So, I think things like that, but I think also kind of getting the message out to other colleges is also something that we could potentially do, because this isn't just a pathology journal problem. This is also, surgery, oncology, clinical journals. So we would need to kind of get this word out to multiple portions of the veterinary community.
I wonder if there's a role for self-publishing open access stuff that's subjected. I mean, we've all done peer review. We've all been subjected to peer review. We all know how to do peer review. Why is there a magical, why are journals magical? In other words, is there a way to sort of have a transparent process like that? I wonder, outside of the traditional sort of journal setup that's trustable, and I don't know the answer.
There's some approaches, not very specific for veterinary pathology, but rather for computer science, where you have open reviews. So you kind of pre-publish your data, and then you have anyone basically can review. And the advantage is that you don't only get 2 reviewers, but a handful of reviewer and a consensus. And the more people the more the consensus will be with the general opinion. So you won't have bad luck with a silly quote unquote reviewer if you have open reviews, and so that's a possibility, but still there's a lot of subjectivity to that to a certain point.
I have some other questions written down from the first lecture in the morning. What can we learn from the, I think it's in-hand, I'm not sure, standardization initiative from colleagues in ToxPath, highly controlled, shorter time frame environment of lab animals versus, quote, messier situation in client-owned animals with spontaneous neoplasms, more problematic follow-up, etc. Does anyone want to start?
Well, I mean, I think we could try to do more prospective studies. That's always the hardest thing to do. But, that would be a way potentially of limiting that variability because, so many of our outcome assessment studies are based on retrospective studies, and these animals are treated, differently because of many, many different, it could be monetary, it could be because of comorbidities, it could be, because of where they live. There are so many things that kind of go into how animals are treated for their neoplasms. So a prospective study would potentially mitigate some of that. But, diagnostic medicine is real life, and there's lots and lots of variability in real life. So I don't think we would be able to create an environment as controlled as some of those lab animal studies, but we could try our best to mitigate some of that variation with prospective studies.
Yeah, so certainly agree that so this is kind of a very similar approach that we're trying to standardize the methods of reporting. And future, we hope that we can improve that. For example, we want to add images for certain aspects. So for example, Rebecca was talking about nuclear atypia in melanoma. That is certainly something not very intuitive, at least for me. And I always have to, look up literature when I report nuclear atypia because I just can't remember it. So for example, we envision that we add images, example images, just as Rebecca did in her presentation for what's low, what's moderate, what's high, so that we can all, balance our cutoff values to a image. That's reporting in our guidelines. So this is something that we hope that we can improve on in the future and give more reference on that.
Yeah, I mean, I think it's very hard to control things, especially on the treatment side, even with prospective studies. I mean, you have an animal that's failing treatment. It's hard for an oncologist not to throw something else, at the animal. And so there's just, and they have the same problem in human medicine too, right? I mean, prospective studies are not easy for many reasons. And there's always going to be that challenge there. And of course, follow up with necropsy. It would also be ideal, but it's also very, very limited in veterinary medicine.
All right, thank you. We have another question. Should we require open source algorithms to be used to process diagnostic samples so that accuracy, methods, reproducibility can be assessed? No black boxes for IE image analysis.
Right, so that probably is, I should answer this. So maybe just for the question before, maybe I can also add, and then I'll go to this question. So for enhance, so I'm not completely familiar with enhance, but what we're planning to do is, and Mike is the expert on this, so we're planning to develop synoptic reports. So basically giving checklists more or less for relevant parameters that you have to, quote unquote, to evaluate for a specific tumor. So we're trying to at least get a minimum list of parameters based on literature, and we're currently investigating how we have to evaluate this literature in order to have a good recommendation. So we want to give basic recommendation for default parameters that need to be assessed in a specific tumor. And then each lab can decide to additionally do Ki-67, for example. But this is also something we are going to look into and also give pathologists hands to easy evaluate literature, so they don't have to look every time into 10 papers to find the parameters I want to assess. All right, so second question, open access, open source, open data. I think that's very relevant. And so we're currently at a state where we are developing algorithms. We're looking into algorithms. We're not using them. So we need data sets to be able to train. We need computer scientists to modify the models and find the best convolutional neural network. So we're at this state where we need to find the best approaches, need to find the algorithms that work. And for this, we need data to be open, and we need to have researchers being to access this. So a big problem is, for example, is that many computer scientists are willing to develop algorithms, but they don't have good data sets. So there's 50 papers maybe on a data set on human mitotic figures that only have 1,000 labels. And so they were modifying the algorithms and increasing the F1 score based on a very small data set. But improving your F1 score by 1% in a data set of 100 labels, this could just be very specific for this data set and doesn't say nothing about the real world. So they had to do it because they didn't have access to bigger data sets, to larger data sets. So I think this is a very necessary thing for improving this field in the future, and especially from an academic point. So for example, our research group always publishes the codes of the algorithms, always tries to publish the data sets. This is very necessary to get this field forward. And from an academic standpoint, I don't think we should hold back information. We need to get this field back on track as fast as possible in order to help us. And publications of codes, for example, are almost required for certain journals, and data publications are very welcomed by many journals. So it's possible, and I think it should be done, especially from an academic standpoint.
Jay, can you hear me?
Yeah, you're here.
Yeah, if you went back to the editor question, how can we get journals to accept studies that replicate data, reproduce data?
Yeah.
I think if it's a combination of what the authors present to the journal, as well as Taryn suggested, we can contact the journal editors and emphasize the importance of it. But there's two things the authors can do. Recognize that the journal wants new novel studies. Try to include something that's new as you validate some of these prior investigations. And even if it's not a new or novel test, tweak one of the other parameters that was used. So if you go in the back of VCGP, you'll see these future considerations where we suggest some things that could be done. So the authors can help the situation. And just like any good introduction to a manuscript that Rebecca or Taryn would write, you can start off in those opening paragraphs with the importance for scientific integrity that studies have to be replicated and duplicated, and cite many of the references that we had in the earlier talk. So I think it'd be approached from two sides. What can you do as the author to ensure it, as well as contact with the editors? Yeah, it'll be a win-win. Good?
Yeah, I have a question, and this is going to be a little spicy, and I don't want to upset anybody, but
I'm out of here.
No, man, you gotta have my back. So, I feel like, at least here, I feel like the ACVIM requirement for publication prior to boarding is responsible for a tremendous amount of garbage in the literature, in the sense of low-end studies taken strictly from records with no pathologist help, et cetera, et cetera, et cetera. And like you say, not giving people credit for being a middle author on a big group paper rather than first author on a very poorly done paper. How do we pressure them? I'll let Don answer that.
I was going to say, the other issue along those lines is AAZV requires something like 3 papers, not just 1 for boarding. So there's a lot of pressure on those poor residents to try and get papers, first author papers out during the residency because they have to get 3 of them.
Yes. Yep. And it seems like they should get credit for, you know, some combination of authorship, but also being involved in a high quality group.
Jay, how would you solve it?
Well, I'd be a dictator, of course. I mean, yeah, I mean, I just feel like the authorship, perhaps the authorship fulfillment could be a more complex algorithm than simply the number of papers and where you are in authorship on that paper. There could be some sort of, you know, more complex calculation that calculates all kinds of things, you know, like the number of animals in the study and the number of collaborators and all of that kind of stuff, you know, that's something almost like an impact factor.
And the editors of those journals are our colleagues as well. So if Taryn and other parts of VCGP will reach out to Steve DiBartola, editor of JVIM, they're our colleagues, and sharing some of the manuscripts that we've read as we put the VCGP together, I think you can lobby to those individuals to say it's time or past time to have a pathologist take a review at these case studies and put that information out on their website so that residents are warned ahead of time. Get a pathologist. What I can't understand from being at the university, why they wouldn't ask a pathologist when there's path data to be reviewed. They must go to parties with pathologists, converse with them. My goodness.
Well, Don, you know, you've been sucked into those projects that turn into a big nothing burger, and you've wasted many, many dozens of hours of your life energy on that. And after a certain point, you get burned enough times that you start saying the no word.
I love your analogies, Jay.
I have another question in the question and answer, which is about necrosis. When evaluating necrosis in a tumor, will you consider collateral necrosis for your total percentage as necrosis produced by ulceration or bacterial contamination? Or are you staying away from those surfaces?
So the recommendation for the soft tissue tumors, at least, is to not count that necrosis as part of your percentage, because it's probably coming from somewhere else other than tumor outgrowing blood supply. So that should not be included, but it's probably not specified for other tumor types. And I don't know, Emma, I don't know if Emily has any additional information to add there. Has that been specified in other grading schemes?
No, I haven't seen it specified anywhere in any grading scheme. So again, another reason why we need to standardize.
Yeah, I mean, that's the kind of detail that we need when describing, you know, how to go about assessing these and reporting out these parameters. If no one has more to say, I'd like to also address the mitotic figure question. So that's a great question. In terms of the hairiness, you're totally right. So the question is, there seem to be some mitotic figures that do not have clear hairy projections, but are still unanimously counted as mitosis. Case in point, that anaphase one from the quiz. So I noticed in a small project we had where we labeled mitotic figures with 3 to 5 pathologists. And then I got nervous when you published your guideline and thought we made an inexcusable mistake. But if I remember correctly, it seems to also occur in the example pictures in the guideline. So totally correct. So the hairy projections are useful. It's not the end all be all of mitotic figures. And so one thing I was trying to get across today is that when you have an anaphase or a telophase mitotic figure, the hairiness is less important because you have those 2 separated chromosome clots. So the lack of hairiness when you have those structures is not, it's not going to change the interpretation of the structure. So you are correct in saying that you can be a little more lenient for the whole hair policy when you're looking at anaphase and telophase. Hopefully that helps answer your question.
Maybe I can add a general comment. So I think generally it's very difficult to give definitions for morphological parameters. So especially mitotic figures. I mean, there's not like a single sentence that can describe what's a mitotic figure like. So that's very difficult, and that's probably also the reason why we do have that. You know, if you remember that recall precision graph where some pathologists were here and some pathologists were here, that's a different interpretation of the definitions or what a mitotic figure is, and it's very difficult to standardize this just by words and even with images. So I think to a certain point we won't be able to get this away. But we're trying pretty hard, and we hope we can, group the pathologies a little bit closer together. And hairiness certainly is a relevant parameter. If you see hairiness, that's certainly a convincing factor. But and we as pathologists, we can integrate so many factors in doing our decision. So, if you don't see hairiness and the whole tissue section is poor quality, you might not call it a mitotic figure. Just because it's blue, but if your other nuclei in the surrounding tissue look perfect and then you have one nuclei it's very blue but not very convincing hairiness, you still might be tending to say, okay, that might be a mitotic figure. So we incorporate many information, and that's what we do as humans and what we're good at as humans.
Hey, Taryn, this is just a suggestion. But get that individual that saw those images to send you multiple images, see what you think of them, get that person to comment on them, as well as it may be very useful to post in your image bank.
Yeah, for sure. Yeah, that's a great point, Don. Feel free to send those to me. And that's our goal with the website too, is to, have it be an educational resource. So we'll have lots of images, of mitotic figures. It's really useful to have the PHH3 so that you at least have a gold standard and something to defend the interpretation that it's a mitotic figure. We're currently working on a mitosis tutorial to kind of go through the features that we discussed today and then have some more images. But the more images, the better. And the more helpful it is, the repetition to be able to get us closer together. As Christoph said, we're never going to be 100%, but, to kind of get us closer together in what we would call a mitotic figure would still be useful for prognostication.
I think a big problem is, so if I can add, so we did publish a publication in Web Pathol this year, I think it was, about the mitotic figure morphology, which is kind of the basic for our guideline also. And so there was a couple of people, of pathologists that came together, and we quite extensively discussed what a mitotic figure is. And there were some discussions on many mitotic figures, and we ended up excluding so many mitotic figures because we just weren't finding consensus. I think the problem really is that, so at least that's true for me, my research was a lot about mitotic figures, so I label a lot of mitotic figures, but I was never really trained what a mitotic figure is. So I started labeling mitotic figures, and eventually I developed a definition that was my definition by just looking at many mitotic figures. So I think that's what happens to a lot of pathologists, that we just look at many cases and eventually define our own criteria, and thereby we just introduce some variability. So probably those images, if we get those to early residents, get a lot of examples, ideally with a ground truth, we can train them with generally accepted criteria, and so they don't have to develop their own criteria. I think that would be variable, especially for early training pathologists.
It'd be really fascinating to blow those things up way big and use eye tracking software to see where exactly we're looking and compare that across pathologists. What are we looking at on those things to make that gestalt decision?
Thanks for organizing all of this.
There's one more question from Kevin Keene, who's the editor-in-chief of ToxPath. And he says, I'll state from the journal ToxPath that we spend no time evaluating the position of authors in the author list. We only look at the quality of the manuscript, submit well-designed studies with high quality and well-validated data with meaningful interpretations, and we will publish it. Yeah, no, I don't think that the comment was about journal editors being the problem was with publication requirements for certain board specialties. There's another one that says, thanks everyone. Should we consider mitotic figure forms specific for each common neoplasm as opposed to assume that a mast cell tumor mitosis is the same as a soft tissue sarcoma mitosis? It's an interesting question.
Hey, Joe, go back to that editor from STP.
Yeah.
It was not the journals and how they might prioritize the authors. It was getting authors recognition at their home base, whether it be at a university or industry and so forth. They're not just the first and the last authors should get the most recognition with however their department chair, CEO, director of the lab establishes a scholarly excellence.
I feel like there's two issues. One is having to publish a paper to get board certification. And then the next is, academic career, tenure and promotion, loops, shenanigans. So I'm curious if any of the image analysis people have thoughts about a mitotic figure form specific to tumors.
All right, so let me answer this question. So I think it really depends on, the same question could go to pathologists. Really depends on what criteria are we using from a mitotic figure. I think the shape of the chromosomes probably are quite similar, but then depends on what do you take as a criteria. Do you also take the shape of the cell? I mean, and specifically for algorithms, we do know for a fact that the tumor type is influencing the algorithm. So we, for example, compared mast cell tumor algorithms to mammary carcinoma algorithms labeled with a very similar approach. And it dropped, although it especially dropped from mast cell tumors to mammary and not the other way around. And this was probably because the mammary tissue was just generally more complex, whereas the mast cell tumor tissue is generally less complex in terms that you just have round cells that are, separated and all that. So, this is going to be very much influenced by the variability in your tissue that you have. But you shouldn't take it for granted that your algorithm for tumor type A can detect mitotic figures in tumor type B, because we don't know what criteria is it using. Is it using the shape of the chromosomes? If yes, probably no influence. Is it using the shape of the cell also? Is it using the color? It's going to influence. I mean, and also for us, we won't have the same performance for different tumor types. I mean, mast cell tumors with granular, we're probably going to miss a lot of mitotic figures. Pigmented melanoma, we miss a lot of mitotic figures unless we do we get rid of the pigmentation, and especially I think the tissue sections doesn't look as good anymore if you bleach. So I'm personally not as convinced with many mitotic figures in bleached tissue sections. Lymphoma, very small, like small cell lymphomas, like when the cell just gets smaller, I'll get less convinced of a mitotic figure as opposed to a giant cell with a typical mitotic figure, which is just jumping at you. So I think there's many tissue-related aspects that just going to influence your performance, both for pathologists and algorithms. Algorithms more so because we do have a domain shift that we as pathologists can adapt to, whereas algorithms just learn statistic from the images and they can't adapt to different situations that they haven't learned.
But I will just say one thing that, looking at image patches from canine tumors versus human tumors, I don't really see that much of a difference in the morphology of the mitotic figure itself. I mean, the cell morphology could be different, sure, but the mitotic figure itself, tends to be the same throughout those different tumor types. If you're just looking at the mitotic figure, would you agree with that?
Yeah, definitely.
And then to answer Jamie's question, Jamie had do you then exclude mitotic figures in normal tissues in the sample? Are you asking about the algorithm? I know that, those are we try to exclude those. I mean, obviously we wouldn't count that in the mitotic count, but how do you deal with those when you get them in the algorithm?
So this actually, I mean, we do know that we're not supposed to count non-neoplastic mitotic figures. And we're trying to do that by avoiding areas that contain non-neoplastic areas, and we just don't count, the fabric sites of mitosis. For algorithms, it's a little bit more complicated. So, for example, for our mast cell tumor algorithm, we just trained it for mitotic figures, and it's just a little bit more difficult to get the second step to train it for cell morphology. And that's because, when we look at a mast cell versus a epithelial cell, for example, we take the surrounding tissue into that consideration. So if you just crop out a squamous cell, epithelial cell versus a mast cell, they might look very similar, depending on the level of the epithelium. But if you see the whole epithelium as a structure, as that no question. So when we take a lot of this into consideration at moderate magnification, algorithms to date usually don't do that. So they usually get image examples of very small, like cell size patches. Although it's technically possible, it's just haven't really been done. So another approach would be to segment the tumor tissue first. So you can, if you only want to have mitotic figures in a tumor section, exclude, hair follicles and all that. You can segment the tumor section, exclude the other area, and just run your algorithm in that tumor area. That would be an easy solution. That's probably more easy to achieve than actually have an algorithm to detect only neoplastic cells.
Yeah, I want to interject this, that in our attempt to standardize parameters, hopefully it's going to help. But I do want to say, Michael Goldsmith, Brian Summers, and a lot of other excellent pathologists certainly didn't need us to help decide what there was a mitotic figure in their counts. But yeah, I think the younger trainees and residents will really enjoy the way that Taryn and others have approached this, and now they can self-train as well as work with their pathologists at home. But having said that, there is this inter-observer variation that we've all talked about, that we've all said is one of the weaknesses. Hopefully just finding the area is going to be one step to bring us closer to reduce that inter-observer variability, repeat these studies, and if you've got outcome assessments, were some of these recommendations by Taryn and Christoph helpful, and you did reduce some of that variability. The younger crowd, I just really hope will pick up on this and do some of those replicating studies.
I just wanted to add too, like we don't want to miss the forest for the trees too. I mean, hopefully, an oncologist isn't going to make a decision on, say, an oral melanoma that has 5 mitoses versus, 2. You need to look at everything else that's going on with the patient and, the clinical presentation, lymph node involvement, all sorts of things like that. And I mean, most of these tumors, they have very, very few mitoses, right? So you're looking at 0 to 1, or you have a lot. So there's only a certain population of cases that are going to be kind of in the gray zone, which will be affected by whether or not, one pathologist calls one mitotic figure a mitotic figure, and the other pathologist says, no, that's not a mitotic figure. Like I don't think you're going to change the prognosis of a huge number of cases anyway, by arguing over, 1 or 2 mitoses. I mean, we certainly need to be all on the, closer to the same page and not making the mistakes like Don said, especially with like new trainees and things like that. I think those papers that you guys have done with the images are fabulous, and they really add a lot of value to especially people who are just learning.
Rebecca, I couldn't agree with you more, especially the fact if you were close to a threshold on the cutoff of a tumor, don't rely on just mitotic count. And your studies with melanoma nicely complement that and provide other means that people can assess. And I'm sure the clinical oncology crowd does not, but there was a time when they put pressure on us to repeat the count and so forth. And I think hopefully we can get those days behind us and they'll look at other parameters than just cell proliferation.
So there's a really good question in the I'm sorry, Don, when you finish?
I think it's also a problem that, we have binary grading systems like below, above. And I think many of us, and including me for a long time, weren't really understanding the statistics behind it, and weren't really questioning the statistics. So, below 5 good prognosis, above 5 good prognosis. That's not true. I mean, below 5 you have a certain sensitivity and specificity of a good prognosis, and above you have that certain sensitivity and specificity of that prognosis. So I think that's something we often don't understand, and we're very addicted to find accounts. When we close to a cutoff, we really like get nervous and like, okay, maybe I have 4, not 5, maybe I do repeat the count and hope to get 5, and then I can say, okay, you have a poor prognosis. That's not the biology behind that, and that's not what your statistics, sensitivity, specificity tell you. And I think especially those cases, maybe it would be useful to have like a gray zone area for parameters where you just need to add your confidence by adding other parameters, by doing some PCRs, by doing immunohistochemistry, whatever. So yeah, especially borderline cases. I think many are just like too often just classified strictly with the cutoff, and also repeating the count. I mean, repeating your count when you have a mitotic count of 4 instead of 5, this is going to give a bias as compared to the original study. They never repeated their count, and you're just going to increase your sensitivity of finding a hotspot when you repeat your count. And this is going to bias you towards having, poor prognosis as what you classify. There's more studies we need to do to understand that effect, I think. Sorry.
So Christoph, that actually segues really nicely into this next question that's in the question Q&A, which is a philosophical question. And I think you guys, it's good to discuss this, which is, do you think that using AI to identify a mitotic hotspot in itself creates bias if humans can't reliably identify these areas consistently and independently without the aid of AI, since the studies which set these mitotic parameters or cutoffs for certain tumor types and grades didn't have access to such technology?
I think the question is a straightforward yes. I mean, as I just said, when we prognostic studies had, based on the patient population, had a sensitivity and specificity of their outcome. But also they had a sensitivity specificity based on the methods that they perform. So again, this is the graph for the pathologists with having a high recall or high precision. They don't have both. So depending on which pathologist you take, you're going to get a cutoff of 5 or get a cutoff of 10. That's ideal for your situation. Same thing for algorithms. So based on our study, at least in our population, you would have a higher sensitivity to actually find hotspots. So generally, your mitotic count, I think it was, your mortality count generally increased by 50%. So generally your values would be, statistically would be higher by 50%. So your cutoff wouldn't be 5 or 7. It would be maybe, 15. So we shouldn't be, so the cutoffs are very much depending on the method that you used and the sensitivity and specificity of that method that you used, of that pathologist that used the method, and of the study population that was used in that study. And changing either one, in this case the method, would change your cutoff probably by quite a bit, I would assume. So yeah, we would need to redo the cutoffs if you use AI.
But also bringing up what Don always talks about, which is that maybe we shouldn't have a number. Maybe we shouldn't have the cutoff be a single number. Maybe it should be a range. And if we look at a range, potentially that will be more helpful and will stop us from being so worried if we get a specific number and then redoing it again because it's close to the cutoff. So having a cutoff that's not a single number maybe is going to mitigate some of that.
So certainly, I think probably especially a gray zone, quote unquote, would be useful, where like the borderline cases where you just have a unacceptable sensitivity and precision that you don't want to have, and you need to find further steps to investigate. The problem is probably from a statistics standpoint, just you can't analyze 10 classes with the one metric. So you can't have a accuracy for 10 tiers. You always have to have 2 tiers to compare accuracy. So you could break down your classes into 10 and then compare 9 versus 1, the other 9 with the other one. But then you would just have very small classes. So then so maybe it's a good approach, but then we would need way more than 20 cases for our prognostic studies. Yeah, I mean, we need to get started collecting cases, right? As a group of pathologists joining up together.
Look, these are fun questions. And if you get outcome assessments to go with the tumor, then people will clamor to lay whatever type of tool they want on top of those cases. But you're going to need the outcome assessments first, because maybe it's not one hot spot. Maybe it's the hot spots that are all through the tumor, the total numbers that are there. You need the data, but what a fun time to do some of these experiments. Several of the people on the panel know, I think it's important to go look at the invasive front. So there's very few studies out there that have compared 4 different techniques with outcome assessment and said, this is the preferred one. And each one of those could vary with various different tumor types. Just because it's melanoma, that information may or may not extrapolate to some of the other tumors. So there's a wealth of potential experiments and ways to look at this that are out there. Good luck. I'm not going to do it.
So I think what that question about the bias and AI really gets a fear of is, are we reaching a point where we're going to diverge and we're going to create sort of standards of care that are out of the reach of some diagnostic laboratories that don't have digital pathology, that don't have access to these kinds of things? Are we creating a stratified standard of care, I guess? And how do we try to ameliorate that somewhat as we go forward? Because, you know, how far are small state diagnostic labs away from having digital pathology and artificial intelligence and all of that kind of stuff, you know?
Jay, don't you think the fact that we don't have third-party payment will be actually a saving grace?
Could be.
Because I do worry about that with standard of care. There's a big difference between standardization and then standard of care. And I've never encouraged that for veterinary medicine in the clinical cases because of where I live, Appalachia and so forth. Some people can't afford that standard of care. And that'll extrapolate over to testing as well. So someone has to choose, someone has to be willing to pay for these tests. So for a change, that could work in our favor, Jay.
Can I also just address something in the chat too that kind of goes with this, which is, you know, requiring future publications to preserve slides digitally and make them available so they'll be available for future evaluation. That's probably not going to be something that the publications can do, but the authors can do that. And that is our idea behind the biobank. This is Don and Fran's idea, and this is basically to accumulate cases with outcome, whether they be whole slide images, ideally blocks, tissue blocks. And that way we could gather, you know, between our institutions, large numbers of cases with outcome data, and we could perform more meaningful studies on higher numbers of cases. So that's our goal, is to you know, use this effort to sort of combine our resources so that we can generate better data that's more useful for all of us.
Aaron, I like your reply, but going back to the point the person made, if at least on the human side of it, if it's NIH funded, all that data needs to be publicly available. And you can request that through the journals. We've never done that as far as I know in VetPath.
To my knowledge, you're not required to provide access to digitally scanned slides, though, at this time. Like, you have to have access, you have to give access to all your spreadsheets and your number data, but you don't have to provide access to the slides.
Jay, can I say thank you for hosting a great seminar and look forward to the end of the month, but I do need to leave if that's okay.
Right.
Well, assuming that was a yes, and I thank all the speakers. I think you did a terrific job, me excluded, but I enjoyed them. Thank you all.
Thank you, Dan. So maybe I can address Jay's comment from earlier. So if you're creating two-class treatment, and that's a comment that Don made a couple of times. I think it's very necessary for us to compare methods. And so there's always going to be different classes depending on whether you have a scanner, whether the patient's going to pay. I mean, we already have it. I mean, if Rebecca is asking for Ki-67 for melanoma because it's a borderline case, some might be able to pay, some won't. So we still have that situation. Same for treatment and all that. So we still have that. But if we have studies comparing the methods, that would be useful so we can decide. So example, if you have a study with the same study population using routine mitotic counts with only manual routine methods with AI, we can look whether AI is better, and then we can say, okay, for AI I use this cutoff, for manual I use this cutoff, and you have different statistics supporting my prognostic outcome. So I think it's very valuable to compare methods. Also, same thing for mitotic count and Ki-67. I mean, if it's a borderline case and you can afford to do Ki-67, good for you. I mean, it's just going to help you find a good treatment. But if you have 0 mitotic figures or 100 mitotic figures, I don't think you need additional costly approaches. But if you have borderline cases, this is going to support your information that you get. So yeah, so it's important to compare and then find also smart combinations. I mean, it's not really smart to combine mitotic count and Ki-67 as a default because they are kind of reporting similar things. But if your mitotic count is inconclusive, Ki-67 might give additional information. So those considerations we need to make to find smart combinations and situations where one or the other parameter might be useful.
Yeah, I mean, I think the same discussion happened when IHC first really sort of came around, right? People were like, well, what if we can only tell things with IHC? What are we supposed to do if we only have H&E? And it's like, well, we can't make H&E better. I mean, we can't make non-AI stuff, be, it is what it is. If it's better, it's better, and the question, I guess, is just how do we, get everybody access to that? And there's so many great, QPath, and they're great free open source image analysis things. I feel like slide scanners are expensive, but scope based slide scanners are not necessarily very expensive. There are some pretty low cost ways to get into slide scanning that don't involve a $150,000 Leica scanner.
Yeah, I mean, you still need to consider like storage of your whole set images and all that. So for routine purposes, I don't accept the big labs. Like we do have a scanner, but we don't use it in Vienna for routine purposes. It's really like trade-off. What's your benefits? And for big labs, I think the biggest benefit is efficiency, so that you can have a pathologist work in a different country or different city and just send the slide. You have resolution, right? You're not increasing your quality of the image, certainly. So it got to be efficiency. And then in the future, you probably can also improve accuracy and all that with image analysis, or at least get it better again in order to become as good as glass slides maybe.
Well, I think if we don't have any other questions, maybe last call.
Well, just the question about the website, what website may host the data? Do you know? I mean, the JPC is housing the biobank, and some biobank material will be on our VCGP website, but I don't really know how much of that, the data would be on Davis-Thompson Foundation website versus VCGP versus JPC. So it would
Mike
be, yeah. Maybe
I was going to say I can probably chip in on that. Davis-Thompson Foundation, we are pretty close to having a viewer for whole slide imaging set up on the website, as well as we have a whole set of slides that have been used for a variety of slide seminars previously. So we're working on getting all of those put together into just a giant repository. So my thought would be for whole slide imaging slides, we would probably put those on the Davis-Thompson Foundation site and then tag them with VCGP or something like that, so that we can have someone go to just, here's the biobank images. And because they've got things like diagnoses and all that associated, and whatever metadata we're interested in associated with them, people could then filter on whatever they're interested in. So if you're looking for all the whole slide images for melanomas, you could pull those up. Theoretically, our storage is infinite depending on how much money we want to pay per month. But the whole slide images are hosted on a site with pretty cheap storage. So we have the ability to scale pretty readily there. So I would see the Davis-Thompson Foundation website for whole slide imaging being kind of the hub for that. And then as far as metadata and photomicrographs or whatever other kind of data we want to put together, we'll just need to figure it out, but we should be able to piece that together. That's not too difficult given the resources we've got right now. So just brief plug, thanks to the Davis-Thompson Foundation for the new website. The board decided that was important to go forward with, and that's given us a lot of new tools to work with in making all this work.
I think it's going to be a very valuable tool to have, the whole slide images accessible for researchers. And also, if this is going along for publication, there's, there might be other sources where you can publish your images if you want to make them available to other researchers. So, for example, we are publishing quite regularly our data sets. So when we develop an algorithm, we usually also publish our data sets. So this could be either done by a data set publication. There's journals interested in data, and those need to be labeled, and they're really interested in those. Some regular journals might also be able to have those as supplemental material. And there's also some software where you can upload freely. For example, GitHub is a software where you can just upload data and then have a link for your publication. So if this goes along with the publication, there might be ways to make those accessible. And this is a citation for you. If someone else is using your data, they have to cite your paper. And this is also helpful for you and for the community, of course, because people can work with your data.
Well, if we have no more questions, since the mitotic figure people are here, I just want to really quick show you. I found what may be the best mitotic figure ever found on anything ever, but we can fight about it. So I clearly think that it's a samurai chopping off someone's head that's then flying off. Pompey thinks it's a smirk, but I think it's just clearly a samurai chopping off someone's head. So.
That's pretty nice.
I think that's pretty clear, Jay. It's pretty
right,
clear.
right?
Right. Yeah.
It's just crystal clear. Okay.
Yeah.
Jay, we need to talk after this.
Yeah, I was pretty sure that was a placenta with a fetus.
Oh, wow. Now that's disturbing.
I saw that too, Emily. I did see that too.
Great minds. Great
One
minds
of us
alike.
is disturbed. I'll just leave it at that. Okay. All right. Well, I think that's about it. Thank everybody so much, and looking really forward to part 2.